Good morning, and welcome to today's webinar on the basics of getting started with base editing. We're glad you could join us as we explore this really next generation and cool trending topic in CRISPR. My name is Jennifer Bennett, and I will be your moderator for this session. Before we begin, I'd like to go over a few recommendations so that we have a smooth and productive session today. First, please ensure your microphone is muted to minimize any background noise. We also highly encourage your participation. So if you have any questions at any point during the presentation, please submit them using the Q&A function in your webinar panel. And we'll address these during the dedicated Q&A session at the end. One note about questions: if you have questions about the use of base editing in your specific workflows or experiments, we encourage you to contact our CRISPR experts to help you set those up, and that information on how to contact them will be provided at the end of the webinar. Now it is my pleasure to introduce our speaker today, Catherine Smith. As our technical support manager, Catherine brings deep hands-on experience and technical knowledge to her role. With over 15 years in the life sciences and 3.5 here at Synthago, she is really at the forefront of this type of knowledge, especially in terms of customer engagement and helping scientists and researchers overcome these challenges in getting their CRISPR workflows set up on a daily basis. Her unique position gives her invaluable insight into the practical applications and nuances of CRISPR technology. And today she'll be sharing some of that expertise with us. So with that, I will hand it over to Catherine to begin the presentation. Catherine, the floor is yours.
Thanks so much, Jen, for that introduction. And thank you all for being with us today. It's my pleasure to talk about base editing and how you could get started with using base editing in your experiments. So let's start with the basics. CRISPR-Cas was first described as a genome editing technology in 2012. And when we think about canonical CRISPR-Cas9 editing, we think about this simple and elegant two-part system consisting of the Cas9 nuclease and the guide RNA. Within this system, the Cas9 nuclease creates double-stranded breaks in the genome at sites that are determined by the sequence of the guide RNA molecule. The first applications of CRISPR-Cas9 genome editing relied on the endogenous repair mechanisms to create genomic changes or edits in the cells. Error-prone non-homologous end joining can be used to create disruptive frameshift mutations, while homology-directed repair can be leveraged to precisely insert or knock in sequences that are encoded on a homology-directed repair template. Within the last 13 years, the CRISPR toolkit has grown with the development of exciting new technologies and methods. For example, nucleases beyond Cas9, like the Cas12a shown here, have opened additional genomic sites to editing through the use of alternative PAMs. Additionally, next-generation CRISPR technologies have been developed to allow for precise editing without the creation of double-strand breaks. Among these technologies, prime and base editing are particularly prominent. Today, of course, we're going to be focusing on base editing. Base editing is a CRISPR dual DNA bases without double-stranded breaks. Base editing is performed using base editors, which are comprised of Cas variants fused to deaminases. The base editor shown here is the first base editor to be developed. This work was published almost 10 years ago in 2016. So how does fusing a deaminase to your Cas enzyme allow for you to make these individual base changes in the genome? Well, the process here starts like most other CRISPR edits. The Cas guide RNA complex binds to and opens the genomic target sequence, and this brings the deaminase into close proximity with the genomic sequence, creating the opportunity for base edits. What happens next depends on the type of base editor you're using. Cytosine base editors include cytidine deaminases. As you might expect based on the name, a cytidine deaminase deaminates cytidine. This results in a uridine intermediate, which is converted to thymidine through DNA replication or repair. Thus, cytosine base editors are often said to convert cytosine to thymine. In contrast, adenine base editors include an adenosine deaminase. The mechanism here is really similar to that of a cytosine base editor. Adenosine deaminases convert adenosine to guanosine via an inosine intermediate. I will note here that there are other base editors in development beyond adenine and cytosine base editors. However, these are less established technologies, so they're not going to be a focus of today's conversation. Regardless of the base editor that you're using, base editing initially creates only an edit on one strand of the genomic sequence, the strand that is not bound by the guide RNA. Very early on, researchers realized that using a nickase as opposed to a dead Cas9 and creating a nick on the edited strand helped bias repair outcomes towards the incorporation of the edit. This biasing of mismatch repair towards the edit, incorporating the edit, of course, increases your base editing efficiency. While this figure depicts a cytosine base editor, this nicking strategy is used for both common base editor types. In addition to the use of a nickase, there have been a number of other refinements to base editors over the years. For example, traditional base editors have an exposed deaminase, shown here in red. Due to the general availability of this deaminase domain, these deaminases tended to come into contact with non-target regions of the genome, causing unintended and unwanted single nucleotide bases at off-target sites. One of the strategies for ensuring that the deaminase only functions at its target site is to literally embed it into the Cas9 architecture itself. And you can see here that the deaminase, shown again in red, is really tucked into the Cas9. This sterically shields the deaminase until the base editor complex has bound the genomic target sequence dictated by the guide RNA. This Cas embedding strategy is the strategy that is used by Synthigo's AccuBase cytosine base editor to avoid off-target base changes when using this Cas enzyme. Now that we've talked about how base editing works, let's talk about why you might want to use it. So first, there are safety benefits to base editing. Editing without double-stranded breaks lowers your risk of unintended genetic changes, such as translocations and other large-scale genomic rearrangements. And it also reduces the toxicity of CRISPR editing, particularly if you're trying to edit multiple sites at once. Many cells poorly tolerate the creation of multiple double-stranded breaks simultaneously, and so using base editing allows you to sidestep that safety and toxicity concern. Base editing is also very precise and predictable. It introduces individual base changes in a predictable way at locations that are very well defined. Unlike a more traditional Cas editing strategy, you aren't depending on cellular repair mechanisms like non-homologous end joining, which do tend to introduce changes that are a bit unpredictable at each cut site. And finally, base editing is relatively efficient, which makes it a practical precision editing strategy for both experimentally and therapeutically important edits. Let's talk about how base editing could be used in your experiments. Here, I'm going to focus on some common edit types created using base editing and the high-level guide RNA design principles for achieving these edits. An obvious application of base editing is to the creation of specific base changes, such as the creation or reversion of disease-associated SNPs. With cytosine and adenine base editors alone, it may seem like your scope is a little bit limited, but it's important to keep in mind that nearly half of all pathogenic human SNPs are caused by G to A mutations and are thus considered addressable by adenine base editors. Another 14% of pathogenic human SNPs are caused by T to C mutations and are considered correctable with cytosine base editors. Base editing can also be used to create premature stop codons, disrupt start codons, and disrupt splice sites. These strategies are typically used for disrupting normal gene expression. In other words, these are base editing strategies that can be used to create gene knockouts. Designing guide RNAs for base editing has a lot of overlap with designing guide RNAs for other CRISPR applications. However, there is one additional factor that you'll want to keep in mind. And this factor is the editing window. The editing window is the stretch of sequence that is the most accessible for base editing when the base editor complex is bound to the target site. The size and position of the editing window is unique to each base editor, and that is something that you will want to account for when you're doing your guide design. For example, the editing window for Synthago's AccuBase CBE corresponds with positions 3 to 12 of the target sequence. But within this editing window, conversion efficiencies are highest at positions 7 to 11. And this is very typical. A lot of base editors do have a bit of a sweet spot within the editing window. And when you're considering guide design for your specific base editor, you should be able to check the characterization data for this specific base editor to confirm the positions with the highest conversion efficiencies within the editing window. With the editing window in mind, let's talk about how you might approach designing guides for each of the editing strategies I mentioned earlier. We'll start with using base editing to create specific base changes. So let's say that this is your dream edit site, and your experimental goal is to convert this ACA threonine codon to an ATA isoleucine codon. In order to create this edit with base editing, you will need to identify guide RNA sequences that place your edit site within the editing window for your base editor. In this case, if you are using Synthago's AccuBase cytosine base editor or AccuBase CBE, you would be in luck. There is a suitable PAM site that places this edit into the editing window. Now, you may be noticing that there are a couple of cytosines here that could potentially be edited. You have your intended cytosine, and then you have this other site nearby. You should expect that other editable bases within your editing window will be converted at some frequency, and these are called bystander edits. Whether or not your experiment can tolerate bystander edits will depend a lot on what it is that you're hoping to do and why. If you're in a situation where your experiment cannot tolerate any bystander edits, you have a couple of options for avoiding these. The first is to design alternative guide RNAs that shift your editing window such that any potential bystander editing opportunities are no longer within the editing window itself. Another strategy is to use clonal screening if you believe that the edit frequency at your target site will be higher or better than the edit frequency at your bystander site. And I think that this clonal screening strategy can be sort of hard to picture, so I've got a bit of an example here. In this case, if we were using Synthago's AccuBase CBE, our desired edit at position 2 does fall within the subregion of the editing window associated with higher editing efficiencies. This bystander site, on the other hand, is within the editing window, but it's a little bit outside of this, ideal subregion. Therefore, in your population, you might expect to see a pattern like this, where the frequency of your intended edit is significantly higher than the frequency of your bystander edit. You could perform clonal screening in this case to identify cells with only the intended edit at the desired zygosity. The success of this strategy, of course, is totally dependent on the edit frequencies in your system and how these edits are distributed across alleles and within cells. Clonal screening can be a challenging and labor-intensive process, but it is a path forward if bystander edits cannot be totally avoided by strategic guide RNA design. Next, let's talk about how base editing can be used to create premature stop codons and disrupt normal gene expression. If you're using this strategy, you will need to start by identifying codons that could be converted to stop codons through C to T base changes. As an example here, we have this CAA codon, which could be converted through base editing to a TAA stop codon. As for creating any other type of base edit, yes. So to broaden your editing options, you can consider creating a stop codon by targeting the non-coding strand. So for example, if you have an in-frame TGG tryptophan codon, you can target the cytosines on the non-coding strand and create a stop codon in frame on your coding strand. Regardless of the strand that you're targeting, you will need to select your guide RNAs such that your intended edit site is placed into the editing window. And so in this case, I'm showing a possible guide RNA design for the creation of a TAA stop codon by targeting the cytosines on the non-coding or complementary strand. Since we're targeting the cytosines here on the non-coding or complementary strand, you will notice that the guide RNA sequence is shown in 3' to 5' orientation, left to right. And so the PAM here and GG-PAM here is shown, backwards on the bottom strand. When you're considering a stop codon creation strategy, particularly if you're looking at editing opportunities on both strands, it is important to keep in mind that the strandedness matters here because base editors only target one strand for editing. The easy way to remember this is to consider your PAM and your base edit as occurring on the same strand. Another strategy for disrupting gene expression is to disrupt a start codon. To identify potential guide RNAs for this edit type, you'll need to start by identifying your start codon of interest. The nice thing about this strategy is that it is compatible with both adenine and cytosine base editors. If you're using an adenine base editor, you can place your editing window to target either the adenines on the coding or the non-coding strand. And if you're using a cytosine base editor, you can design your guide RNAs to place your editing window over the cytosine on the non-coding strand. In this case, I'm showing an example of a guide RNA design that targets the adenine of this ATG start codon using a adenine base editor. While you can perform this type of edit using either an adenine or a cytosine base editor, many publications showing the strategy do use an adenine base editor, I think largely because there are a couple of different opportunities for editing. The final gene disruption strategy that we'll be discussing today is a splice site disruption strategy. And this strategy typically leverages the fact that most human protein coding genes have a conserved splice donor site, the GT shown here, and a conserved splice acceptor site, the AG shown here. Again, the nice thing about using a splice site disruption strategy is that you can use either an adenine or a cytosine base editor to target either the splice donor or the splice acceptor. If you're using an adenine base editor, you can target the adenines that are present either on the non-coding strand of the donor or the coding strand of the acceptor. And if you're using a cytosine base editor, you can target the cytosines that are present on the non-coding strand of either the splice corresponding to either the splice donor or the splice acceptor. Again, you'll need to confirm that your editing window falls over your intended edit site. So if we go back to the splice acceptor site we looked at a couple of slides ago, we identify this potential cytosine here on the non-coding strand as our target. We can select a guide RNA design compatible with Synthago's AccuBase CBE that places the editing window over this cytosine. Again, because we are performing our editing here on the non-coding strand, you'll notice that the guide RNA PAM sequence are presented here on the bottom strand in 3' to 5' orientation. So at this point, when we're talking about strategies for creating knockouts using base editing, you may be thinking that these gene disruption strategies seem like kind of a lot of fussing around for something that you could do using a standard SP-Cas9 approach. And it is important to remember that there are some drawbacks to multiplexed edits using Cas9. So the first drawback is genomic instability. If you're creating multiple double-strand break sites simultaneously, you're creating the opportunity for large-scale genomic rearrangements like those shown in this figure. As I mentioned previously, double-stranded breaks also tend to be pretty toxic to many cell types. So if you're performing a multiplexed edit, you may find that the survival and proliferation of your cells is really negatively impacted post-editing. As an example, let's walk through a multiplex edit performed using AccuBase CBE in primary T cells. This multiplex edit was designed to knock out 3 different genes using a mix of splice site disruption and premature stop creation strategies. The first gene, PDCD1, was disrupted by base editing a splice donor. This results in a single gene knockout efficiency of upwards of 80%. The second gene, TRAC, was disrupted by editing a splice acceptor site. The efficiency of this single gene knockout is even higher. The third gene, B2M, is disrupted through the creation of a premature stop codon. This also delivers a single gene knockout efficiency of about 80%. I want to note here that some of our earlier guide RNA design examples were not theoretical. The guide RNA design that I showed as an example for splice site disruption and the guide I showed as an example for premature stop codon creation were actually the guide designs that were used to deliver these highly effective knockout edits in primary T cells. When you multiplex these 3 guide RNAs together, you find a multiplex knockout efficiency of upwards of 70%, which is really amazing for a 3-gene knockout that is performed, simultaneously. It's also worth noting that the edited cells tolerate AccuBase multiplex edits very well. This figure shows cell proliferation post-editing. The cells edited with the cells exposed to multiplex AccuBase edits, shown here in the green bars, display good growth post-edit. This growth is similar to the growth that you observe in an unedited or wild-type control population, shown in the dark bar. In contrast, cells exposed to a multiplex Cas9 edit, shown in gray, performed relatively poorly after editing, and they exhibit, a relatively severe growth defect. So if you're starting to think that base editing might be a good fit for your genome editing needs, here are some things to consider as you get started. As you likely noticed, a lot of your guide RNA design considerations are going to depend on your specific base editor. So how do you make sure that your base editor is a good fit for your needs? As for any CRISPR edit, you're generally going to want to start by thinking about your editing goals and the base changes that would allow you to accomplish these goals. When you're starting to look at specific editors, I really do encourage you to keep an open mind. Not all edits are possible with every editor, and having multiple strategies in your toolkits maximizes your chances of being able to perform your edits of interest. Once you have some editor candidates in mind, you'll want to consider the specific attributes of these editors. For example, does your base editor have PAM sites where you need them? What's the editing window of your base editor? Is your base editor efficient? Is it specific? What format is your base editor available in? Will you be delivering your base editor as a protein, as an mRNA, as a plasmid? These considerations tend to really inform your delivery strategy. And then finally, if you're interested in therapeutic development with a base editor, you may want to consider the licensing of that base editor and how that might impact your ultimate therapeutic development plans. The other key piece of getting started is picking some guide RNA designs. We've obviously spent quite a bit of time on the design strategies for certain edit types, including these, but once you're ready to get started, there are some really excellent free tools available online to support your guide RNA design needs. As a starting point, I am especially fond of BE Designer. I think that it is very user-friendly. It's got some very nice customization options, and it provides helpful visualizations of your edits as well as some other good output data, including predicted off-targets. I also want to flag Splicer or SpliceR, which is a free online tool developed by the Moriarty Lab at the University of Minnesota. This is a tool that is purpose-built for designing guide RNAs for a splice site disruption strategy with base editing. So if you think that you might be looking at doing this sort of guide design, I really recommend taking a look at this tool. Regardless of the tool that you choose, here are some evergreen recommendations for guide RNA selection. So first, you will want to confirm that your guide RNAs are specific. You will typically start doing this by ensuring that there is limited sequence homology between the sequence of your guide RNAs and non-target sites in the genome. I also recommend that you consider the different transcripts encoded by your gene of interest. Particularly if you're interested in using base editing to deliver a knockout, you will probably want to ensure that your disruptive edit targets all of the known protein coding transcripts for your gene. This will ensure that you have a complete functional knockout versus only knocking out some of the protein isoforms. And then finally, where possible, I do recommend that you try a couple of guide RNA designs per edit. All design tools give suggested information about, the predicted activity of individual guides. However, these predictive scores may or may not reflect what you see in your system. Testing a couple guides in parallel will give you some options to move forward with in terms of, potential guide candidates. Once you've completed some edits, you will need to analyze your base edited cells. As for other CRISPR edit types, we recommend a two-pronged strategy for analyzing editing. First, you'll want to understand the edits that are present in the genome, and you'll also want to confirm the functional or phenotypical consequences of these edits. Genotyping via sequencing is the most direct way to assess editing in your cells, and so we will always recommend that you genotype by sequencing to understand what's happening at the genomic level. As shown in these Sanger sequencing traces, this strategy allows you to compare your unedited and edited populations and see the edits that are present or not as a result of your base editing attempt. Sanger sequencing, as shown here, is fast, accessible, and very affordable. But if you prefer to use NGS for greater sensitivity, there are analysis options for both sequencing strategies. I'd like to emphasize here a couple of free tools that are available for genotyping analysis. If you're planning to use Sanger sequencing, I recommend Editor or EditR, which is another wonderful tool provided by the Moriarty Lab. This is a purpose-built tool for base editing analysis based on Sanger sequencing data that provides information about the predicted base editing present in your sample. If you prefer to use next-generation sequencing, CRISPR-SO2, a tool provided by the Pinello Lab, provides NGS analysis for a range of CRISPR edit types, including base editing. And this also provides information about the frequencies of different edits present in your sample based on the sequencing data. Regardless of your sequencing method, you'll probably want to back up your genotyping data with functional or phenotypic assessments of your edited cells. The ideal assay here is going to totally depend on your experiment. If you're trying to perform a knockout using base editing, you may want to use a strategy like Western blotting or flow cytometry, as shown here, to demonstrate that your target protein is actually depleted in the knockout cell population. So today we've talked a bit about how base editing is safe, precise, and efficient. We've also talked about some of the edit types that are possible with base editing beyond individual base changes. As you're probably seeing, base editing is a really well-established technology at this point, with a lot of free tools available to support your needs if you're looking at getting started. I think the best demonstration of the maturity of base editing as a genome editing technology may be the number of base editing therapeutics that are currently in clinical trials. For example, BEAM-201 is the first base editing therapeutic to enter clinical trials. It began clinical trials in 2020, and it actually uses a multiplex knockout strategy for the production of a CAR T therapy. Verve 101 is the first ever in vivo base editing therapeutic. And this clinical trials for Verve 101 began a few years ago. This therapeutic has currently been supplanted by a bit of a next generation therapy, which is also quite exciting. And then finally, Base Therapeutics 510 is the first base-edited natural killer cell therapy to receive IND approval. This received IND approval about a year ago. We are particularly excited about NK510 because this therapy is produced using AccuBase CBE, which is a base editor that we currently offer in protein format. You're with us today, so we know that you're interested in base editing, but if you're interested in learning more and doing some deeper reading, we recommend this resource on our website. If you think that you're ready to get started with AccuBase CBE, you can contact our sales team for quoting at this URL. Finally, thank you so much for joining us today. As Jen mentioned, we would love to take some questions.
Thank you, Catherine, for that excellent presentation. Those are really invaluable insights, especially for our audience looking to get started with base editing. So we have received a number of questions during the presentations, and we're going to move into our live Q&A session. As a reminder, if you have a question and you haven't submitted it, please enter it into the Q&A box, and we will do our best to address it. There are a number of questions that are similar to each other, so I am going to kind of group them together. So if you don't hear your specific question called out, it might be because somebody else had a similar question as well. Okay, so I would like to start, Catherine, talking about, you mentioned base editing being very precise, not needing to create double-strand breaks, but you also mentioned that it can create indels. So how is this possible if they're not actually creating double-strand breaks?
Yeah, that's a great question. So, when you're using a base editing strategy, you are typically relying on mismatch repair to either introduce the edit that is present on the strand of the genomic sequence that's not bound by the guide, or using mismatch repair to restore the original sequence. However, there are other nick repair mechanisms that can come into play, such as base excision repair, and these can result in small indels at the base editing site. However, with most base editors, the frequency of these indels is relatively low compared to the frequency of the intended edit.
Excellent. Okay. So let's see. Our next question, let's talk a little bit more about delivery of the base editor itself. You've mentioned RMPs, mRNA, and plasmid. Can you give a little bit more information about what to consider with delivery and also what format the AccuBase actually comes in?
Yeah, that's a great question. So I'll start with part 2. AccuBase is currently available in protein format, which means that you are going to be delivering that as an RNP. So pre-complexing the AccuBase with your guide RNA and then transfecting that into your cells using typically an electroporation or liposuction-based approach. Now, there are base editors that are available as plasmid or mRNA format. The specific transfection parameters are different depending on your cargo type, but in a lot of cases, you can introduce both mRNA and plasmid format base editors via either liposuction or electroporation as well.
Okay, let's see. The next question is around analysis. So you mentioned a couple of different online tools, or excuse me, for designing. You mentioned a couple of online tools for designing, and then analysis. Of course, you mentioned Sanger and NGS. A little bit of a two-part question. Sanger versus NGS. Is there a preference for one or the other, especially with base editing? And the second part is, can ice be used to do the analysis?
Yeah, those are two great questions. So when it comes to selecting Sanger versus NGS, I think it's really going to come down to your comfort, the methods and tools that you have available to you, and your tolerance for insensitivity. So, NGS is typically going to be more sensitive than Sanger sequencing. That being said, Sanger sequencing is often good enough for assessing base editing in most experimental settings. If you think that you might be interested in pursuing NGS, but you're not certain, an option is always to use Sanger sequencing for early pool analysis and then doing NGS confirmation of any clonal populations. And that kind of gives you the best of both worlds because you're able to do your early screening using a method that's very, very quick and inexpensive. And then you have the confidence of NGS when it comes to the cells that you might actually be using for your critical downstream experiments. ICE is not really intended for base editing analysis. So, ICE is a tool that was developed by Synthigo. It's now hosted by our sister company, EditCo, and we have never validated it for base editing. You may be able to trick ICE into returning a result for your base edited samples, but we can't vouch for the reliability or lack thereof of those results. Editor is really user-friendly. I would recommend that you check this out as an alternative. They also have some really beautiful instructions for using this tool on their website that will be useful if you're unfamiliar and not totally sure where to start.
Perfect. Okay, next question. There have been a number of questions about optimizing the base editor, where to choose the windows. So, for example, one question is, what are the risks of choosing the wrong base editor? And another question is about the opposite strand, right? So if you're changing a base on your target strand, but if you have a gene on your opposite strand, what are some things that you need to look for so that you make sure you don't disrupt genes on the complementary strand?
Okay, yeah, these are good questions, and they're a bit complicated. So I'll do my best to give sort of concise, high-level answers. When it comes to selecting your editor and placing your editing window, a lot of these questions probably need to be answered with experimental testing. So, every base editor is unique. We can really only speak to AccuBase CBE at this point, but you'll want to make sure that on paper, your base editor has pretty good performance in terms of the edit specificity. Some base editors have smaller editing windows, which if you're really concerned about bystander editing might be helpful. And then ultimately, again, this is a place where you're probably going to want to design a few guide RNAs per base editor and test them and see what happens. I wish I could give more conclusive answers, but the bottom line is there's never any substitute for testing it in your cells. When it comes to edits that are on opposing strands, so it sounds like in this case, we're talking about a situation where you might have overlapping genes on each strand. Is that accurate, Jen?
Yes.
Okay. Yeah. So, with any edit type, if you have a gene that's encoded in one direction on one strand and another gene that is encoded overlapping on the complementary strand, it can be very difficult to place your edits such that you're only impacting one gene. So base editing starts by creating a mismatch, but that mismatch is resolved as a result of mismatch repair, right? And so you would expect that any edit that you make on one strand will be represented on the complementary strand as well. You may be able to get fancy with your guide design and place the edit such that it falls in an exonic region on one gene and in an intron on another gene. But again, the best way to set up that sort of editing is going to be highly specific to your experiment and sequence of interest.
Perfect. Thank you, Catherine. I know that's a complicated question, right? In the optimization, even when it's not just base editing and the traditional Cas9 editing, there's still a lot of optimization to consider in those early stages.
Yeah, absolutely.
Let's see here. Another question is, any tips for multiplex base editing optimization? Clearly, you said it was very good for multiplex because of that lack of double strand breaks. So if they're looking to do multiplex editing, maybe how should they go about structuring those early experiments?
Yeah, absolutely. That's a great question. So, when you are considering any sort of multiplex edit, I do recommend starting out by identifying guides that work very well individually. And this allows you to, for any given edit site, have a couple of different candidates that you test and then, pick the one that works best. And at that point, once you've identified your lead candidate for each individual site, you can start experimenting with combining guides. When it comes to multiplex editing, again, unfortunately, there's no substitute for testing these things experimentally. Certainly multiplex delivery in this sort of setting is relatively straightforward because you're typically going to be looking at a system where you mix your guides, you complex your guide pool with your nuclease, and then you transfect that if you're using an RNP approach. I think unfortunately, again, this is a case where a lot of optimization is required, but my single biggest piece of advice would be to do the upfront pilot work to identify your best individual candidates before moving forward with multiplexing.
Yeah, that makes a lot of sense. Um, and so we did have one question come in around the CBE version we sell. Do you mind just giving a quick, brief overview of the AccuBase again for those that might have missed it?
Yeah, absolutely. So Synthigo's AccuBase CBE is a cytosine base editor with an SP-Cas9 nickase component. And so this means that AccuBase CBE uses an NGG PAM, and it uses guide RNAs that are very similar to guide RNAs that you might use for a standard Cas9 edit. Of course, the specific targeting sequence may be a bit different. We currently offer AccuBase CBE in protein format. And it is, as you can see from the data that we showed, it does have really excellent editing efficiency, assuming robust guide selection. And it also has very favorable off-target profiles. So I haven't shown this data, but the incidence of indels at the edit site is relatively low with AccuBase CBE. And it also introduces off-target edits related to, like bystander editing type phenomena very, very rarely. And so it's a really, really strong nuclease, both really, really strong genome editor, both for research applications, but then also therapeutic development.
Okay. Let's see. What about screening? Is base editing a good idea to be used in a screening platform? Are there challenges, limitations as a screen?
Yeah, so base editing has been used for screening approaches before. It depends on the kind of screening you're doing, but a lot of CRISPR-based screening approaches do use a delivery method that allows you to deliver really large pools of guides. So something like a plasmid or a viral delivery mechanism. So, it certainly can be used. You would need to design guides that allow you to accomplish your specific screening goals, such as, splice disruption or premature stop creation for knockouts, which I believe is typically how base editing has been used for screening in the past.
All right, now here's kind of a big one. Um, could you talk a little bit of the comparison between base editing and prime editing and when one option might be preferable to the other?
Yeah, that is a great question. So, one of the drawbacks of base editing is that you really can only create these individual base edits, right? And so if you consider all of the base edits that are possible using cytosine and adenine base editors on both strands, right? You're looking at being able to create 4 of the 12 possible base changes. And with prime editing, you really don't have this restriction because you can encode your specific desired edit into the prime editing guide RNA. You can create any base change that you have in mind. Prime editing also allows you to install larger edits, right? So you could use prime editing to insert a small tag in theory. So, I think that when you're comparing base editing and prime editing, I would let your editing needs take the lead. If you're interested in just creating a single base change that can be created with cytosine or adenine base editing is probably going to be a little bit more straightforward from a guide design standpoint. And there's going to be less guide optimization involved because the guide is simpler. That being said, if you have a bigger edit in mind, prime editing is simply going to be the better way forward because those edit types are not possible with base editing.
Yeah. Well, and there's also there's the availability, right, of prime editing tools as well. And I think, correct me if I'm wrong, but the base editing tools have been around for quite a bit longer, and they're a little bit further advanced into that commercial space, tested with further evolutions for increasing efficiency and fidelity, whereas prime editing is still a little bit early, and more optimization might be required for a prime editing strategy.
Yes, those are good points. And thank you, Jen, for adding those. That's also true. Base editing is just a slightly older, more mature technology, which means that there are more resources available.
Let's see. Next question. Does AccuBase require a license for commercial or therapeutic use? And how could Synthigo help a customer go about either acquiring that license or learn more information about it?
Yeah, so that's a great question. And that is certainly something that we would be happy to discuss with you. We do offer AccuBase for, commercial and therapeutic use through, it would be require a license. And we would recommend that you contact our team to talk a bit more about your specific situation and how we could help.
Yep. Okay, let's see. Oh, there's a lot of questions here still coming in. Okay, let's go back to one about. So, several of the questions that we've gotten so far are about kind of putting it into their system, working on specific mutations, promoter regions, splice sites. So, just in general, can you talk a little bit your role at Synthigo and how you might, as these people, if they were to reach out, how what you would do to help them or guide them in further optimization, post this webinar?
Yeah, absolutely. So I am the manager of the technical support team at Synthigo, which means that a lot of my role is working with researchers who are interested in using our CRISPR editing tools in their exciting experiments. And this means that if you've got, detailed specific questions about how, these design tools or AccuBase CBE could be used in your research, we would be happy to help you and provide some suggestions that are tailored to your specific experiment. Our contact information is on our website. And I think that, if you've got specific pressing questions about how you might use AccuBase CBE, it's probably best that we handle those one-on-one so that you get exactly what you need to move forward.
Yep. That's great. Yeah, and I will brag a little bit here on Catherine. She does a fantastic job working with customers to really get them optimized and troubleshooting through their experiments. Many of our customers brag about her and are very grateful for her dedication to providing resources and guidance. So, if you have a specific question and you've put that into the Q&A, we would be happy to discuss that further with you if you contact our sales team. The last question I have now, Catherine, is kind of a broad question. Can base editing be used in non-human or non-biomedical systems?
Yeah, that's a great question. And thank you, Jen, for bringing that up. So yes, base editing has been used very broadly across a range of systems within, the biology discipline broadly. Base editing has been used successfully in really all of our most favorite eukaryotic model organisms. So, yeast, C. elegans, Drosophila. Base editing has also been used very successfully in plants, including, the model organism Arabidopsis, of course, but also in many plants that are crops of serious agricultural significance. So, for example, base editing has been used in really all of the major cereals, rice, corn, rice, maize, wheat with good results. Um, I personally think that some of the most exciting applications for base editing outside of biomedical research are in the crop development sphere. There's been some interesting work showing, for example, that you can use base editing to introduce herbicide resistance traits into rice, which is an important agronomic trait if you're otherwise using that herbicide for weed control. So, base editing was first published in 2016. At this point, it's still, a relatively new technology as we enter the second decade. Right? It's going to be really exciting to see the applications for base editing across biology.
Awesome. Yeah, I had no idea that it was used in so many different crop products. I really thought it was healthcare based because of the precision and the double strand breaks.
Yeah. Yeah. So, as far as I know, none of these things are being grown commercially at scale. But again, we'll see what the next decade brings.
Well, it seems like a pretty versatile tool at least, despite some of the limitations when you originally think about, making a single SNP change. So
Yeah.
thank you, Catherine, for your great presentation. Thank you all for attending, for your really insightful questions. If we weren't able to answer your question live, please know that we will be in touch to get those answered, and feel free to reach out to our experts at any time with further questions about AccuBase, AccuBase licensing, or base editing optimization. Again, thank you for your attention today. We will be making this webinar on demand on our website, and we'll be sending out that link in an email following this webinar. So thank you again, and enjoy the rest of your day.
Thank you.